you look at the slit of yellow light that was made up of this mixture of red and green, what you saw was the front edge of it, the leading edge, was reddish, and the trailing edge of that yellow slit was greenish, with yellow in the middle. Now, that itself is fine, and folks who believe in that standard model of color vision would say, well, that just means that the red cones must have a faster time constant and turn on faster, so you see red faster, and green cones have a slower time constant and see green later, so that explains why red leads and green trails and is yellow. Now, if that was true, the same thing must happen with a yellow made up of pure yellow, because because of the way they're exciting the so-called red and green cones. But when he did the experiment, what he found was that as you move the yellow light that way, it does not split up that way, but stays a pure yellow. That's a result that flatly contradicts the three-cone model of color vision. It completely invalidates that model.
What does pigmentation have to do with any of this? You talk about, in the early part of your book, the distinction between three cone types and three pigments are not necessarily synonymous concepts. What is this about? Why is this so important to your whole theory?
Well, I mean, certainly the pigments that are in the receptors are very important, because that's how light is detected. That is, as light enters these receptors, there has to be something that catches the light and basically releases a mechanism that converts it into an electrical signal that eventually goes on to the brain and says, hey, I detected some light here. So that's what these pigments are. In terms of the standard model, they visualize that each of three different types of these pigments with sensitivities in the red, green, and blue must each be sequestered in each of three different kinds of cones so that they can separately generate a red, a green, and a blue signal. Now, in terms of the model I'm talking about, there is no hard and fast requirement that the pigments have to be distributed any particular way. The model is actually fairly indifferent to how you put the pigments in there. All it needs is that there must, of course, be pigment to convert and to detect the light and convert that detection into an electrical signal that the brain can see. And that, in fact, is another critical part of the model that I haven't addressed. I mean, the way I describe the model is talking about how light is spread along the length of the cone, but there's a key, another step that's key, that information about color spread along the length, that it has to be converted into the information that the brain itself can interpret as color. That the job isn't finished there. And that's the critical sort of timing information that I can talk about, if you like.
I have a few other questions, though, before we go to that. Yeah.
Sure.
I want to know for the public why you went into atomic physics, what you were doing in the defense industry for the last 25 years, how you got into lasers. Share a little bit about you. Where'd you come from?
Okay.
People want to know who you are. Where'd you come from? Who is Dr. John Medeiros? Is it Medeiros?
Medeiros, yeah.
Medeiros.
Sure. Well, I mean, I was actually since whenever I was a little kid, I was always interested in physics. And I took up that study. I went to the University of Massachusetts, and I wound up studying atomic physics under a superb tutor there, Dr. Robert Krakoff, and I really very much enjoyed atomic physics. I got to play in the lab, or I got to play with some very sophisticated equipment and tinkered with matter at the sort of deepest levels, and I really, really did enjoy that. But I also had a feeling, too, at the time, by the time I finished, that I really wanted to do something a little more expansive, and I was really kind of always interested in biophysics. And I had the opportunity after graduate school to do a postdoctoral study involving laser damage mechanisms in the eye. And that's kind of how I got started in this whole thing, because although I knew about the physics and lasers, I didn't know really a lot more than sort of very vague things about the eye. And so part of the process when I went on to this postdoctoral studies was to learn as much as I could about the eye. And that was the fascinating thing, because I came at it from sort of a point of view of, I wasn't taught, I guess, the standard doctrine about how this works and that works, and I had to try to figure that out for myself. And I may not have gotten it all right all at once, but I got to the point where I got to thinking about this model eventually and say, well, why not this model? From the point of view of a physicist, this kind of thing makes a whole lot of sense. And from the point of view of, of just how we see it in terms of the way people generally look at the eye is not so necessarily so obvious. That's sort of how I got started. And I continued some of that studies later through another research associateship at University of Western Ontario, and then later when I was a professor at Penn State. And then later on, you asked about the defense industry. That was because at one point I got a better offer, as it were, or offer I couldn't refuse, and wound up, one of those. And got into doing some studies out in wound up being some secret laboratories where we actually had to play with some fun equipment there too. But that stuff that involved lasers and optics, which was things that were near and dear to my heart as well. And so in the next 30 years, I wound up playing in that field.
Okay, so now I want you to explain to us the distinction between a biophysicist and a physicist. What is biophysics? What is physics? What's the distinction?
That's not such an easy question. I'm not sure that's such a good distinction. Well, biophysicists, I think the difference is that they try to apply physical principles to the understanding of biological systems. That is, taking what we know about physics. Physics is kind of the study of basic aspects of energy and matter and how things interact, and apply it to, in fact, what's a very hard field. Biological systems are, in fact, very complex systems. And, physicists in general like to sort of treat things in terms of simplify them to their the lowest common denominator, and treat them in sort of a theoretical fashion that you know exactly what's going on. And that's hard to do in terms of biological systems because they're just complicated. I mean, they're wet, and they have all kinds of things going on with them. And in part, that's kind of what makes them interesting. They're not simple, and it's hard. A hard part of the job is trying to figure out how to simplify it enough that you could get your arms around it and understand it and understand what's happening. That's what you try to do in physics, is simplify it to the level where you can understand the model of what's going on. So I guess one of the attractions maybe of biophysics is that, in fact, it's not easy, and that's certainly been the case in terms of vision, that there's an awful lot going on. It's not trivial, but it's in fact a very interesting system. That's one of the things I got to have as a real appreciation of the time I spent studying the eye, is that it's an absolute engineering marvel, and whoever designed it sure knew what they were doing better than most physicists off the street might have. I mean, it's a extremely elegant design and works extremely well.
When you and I first spoke by phone and I heard about your book and your work, you shared with me that you had written some of the experts in the field in the area of vision with your new theory and model. I want you to share what happened to you, because what happened to you is very common, believe it or not. Share it.
Well, there's a couple aspects. I mean, early on, when I first had this concept, when this concept first came to me, I got kind of excited about it. I thought, well, surely somebody's thought of this, because this, it must have been done before, and for some reason it's been rejected because somebody thought about it and found out it couldn't work. And as I looked into it more and more, I found that, in fact, it did work. And so I got real excited, and I wrote a paper that I submitted to the most prestigious journal I could think of and tried to send it in. And with their great anticipation, I was thinking, here we go,